Okay, hello friends, welcome back to my channel Medicine 101. Today we'll talk about hypersensitivity. Okay, basically I will just focus to type on hypersensitivity. Before starting the type on hypersensitivity, I'll give you the very basic or the introductions idea about the each individual hypersensitivity. Now, whenever we are just talking in terms of the hypersensitivity, what do we mean by hypersensitivity? Okay, so hypersensitivity basically from the term it's quite easy to understand. Whenever we get the overreactions of the immune system, just like this. But in a word, in a nutshell, we can say that whenever we will get the tissue damaging reaction, that is known as what, the hypersensitivity. So the tissue damaging reactions, so that is known as the hypersensitivity reactions. Okay, so let's write it over here that. Hypersensitivity we can define in terms of tissue damaging reactions. So tissue damaging reactions. Whenever the tissue will get damaged, after that what type of the reactions we'll see, basically you can see that is the what, hypersensitivity. Now on the basis of these things we can classify the hypersensitivity the four types: type 1, type 2, type 3 and type 4. We'll talk about each of them individually, okay, in details. So first today we'll talk about type 1 hypersensitivity. Now we can classify hypersensitivity into four types. Now see, we can classify the hypersensitivity in terms of the type 1 hypersensitivity. Hypersensitivity reactions over here. We can classify in terms of type 2 hypersensitivity reactions. We can classify in terms of type 3 hypersensitivity reaction, and we can classify also type 4 hypersensitivity reactions over here. Okay, now see, how can we just differentiate each of the other classifications? I mean, how can we just differentiate, or you can separate the type 1 hypersensitivity from type 2, and how can we just differentiate the type 2 from type 3 and type 3 from type 4, just like this. Okay, so we can write it over just like this. We are using the Roman form. Okay, Roman letter, yes. Now see, whenever we are talking in terms of the type 1 hypersensitivity, this one is what, antibody mediated. This is also antibody mediated. This is also antibody mediated. So that means the first three hypersensitivity types, that means the type 1, type 2 and type 3, they are what, antibody mediated. There we see the presence of antibody over here. Okay, antibody mediated will get over also here, the antibody mediated. But what about the type 4 hypersensitivity? The type 4 hypersensitivity, that is the what, cell mediated. Okay, that is the cell mediated hypersensitivity. Cell mediated. Now see, whenever you are talking in terms of the type 1 hypersensitivity, in these cases what type of the antigens will get? If you talk about in terms of the antigens, the antigens that will be what, the free antigen and then the foreign antigen. Free and foreign antigens. For example, we can see in terms of the pollen grain over here. I'll talk about in these things in details. So for example, the pollen grains we're getting over here. Okay, and what type of the antibodies will get over here? The antibody will get always, that is what, IgE. You get the antibody that always IgE. Now the antibody, where can we get the antibody? This type of the antibody will get on mast cell, of course within the tissue. This Ig antibody will be what, will be seen on the surface of the mast cell in terms of the tissues, and we can also see in terms of the basophil in the circulation. Okay, and we have to know these antibodies in type 1 hypersensitivity, these are fixed antibody. Okay, and the antigens in type 1 hypersensitivity, these are the free antigens. Okay, now let's talk about type 2 hypersensitivity reactions. Very basic things. So what do we know in terms of the type 2 hypersensitivity reactions? So at first, if we just talk about in terms of the antigens, these antigens that will be always intrinsic antigens, and these antigens are fixed. These antigens are fixed antigens. Okay, And what type of the antigens? For example, if I just say you that, for example, this is the what RBC, and within the RBC we can get intrinsic antigens. That means the antigens within the body. Okay, that will be from within the body. Now here we will get what type of the antibodies we will get over here. If we talk about in terms of the antibody, we will get IgG or IgM. We will get IgG or IgM. These antibodies will come over here and they will do the reactions further. Okay, we'll talk about in details in the whenever I'll discuss specifically in terms of the type 2 hypersensitivity, where we will get in details about it. Okay, and see, in these cases the antibodies, this is not free, this is not fixed. Okay, so antibody, that is what, free form. So the antibody that will be in the free form. Okay, the what, in these cases we are getting the antigen fixed. In the first cases we are getting the antigen free. Okay, antibody fixed. In these cases we are getting antibody that is fixed, and the antigen that is fixed and antibody that is free. Okay, the reverse case. What about the type 3 hypersensitivity? In type 3 hypersensitivity, how can we define? In these cases, if we just talk about in terms of the antigens, the antigens can be intrinsic or extrinsic. So let me write it over here. This can be extrinsic antigen, that can be intrinsic antigen as well. Okay, extrinsic or intrinsic. Now in these cases, the most important thing over here, that the antigens and antibody, the antigens and the antibody, both of them are in the free situations. That means they are freely movable within the circulations. So here we get what, the antigen and antibody, both of them are what, free. Both of them are free. Okay, and whenever they are free, whatever thing we'll see, the antigen and antibody within the circulations, they will combine with each other and they will form the antigen antibody complex. So in the main classical, the main characteristic features, the main characteristic feature in terms of the type 3 hypersensitivity reactions will get, there will be formation of antigen antibody complex. That's the main features over here. So we'll get over here in these cases antigen antibody complex formation. After that, they will get deposited on the basement membrane. For example, in the glomerular basement membrane, many tissues, others also as well. Now, what type of the antibodies we will get over here? We will get antibody over here, just like this. This one, like in terms of the type 2, the same type of the antibody we can also see. We can get IgG, we can get IgM antibody over here. Okay. And what about the type 4 hypersensitivity? Type 4 hypersensitivity can classify in terms of the cell mediated. That means, for example, if there is any type of the viral infections or the fungal infections, there will get the activations of the T cells. That means T lymphocytes over here. So, for example, if we just say in terms of the viral infections, in terms of the viral infections or the fungal infection, in these cases we will get the activations of the T cells. Okay, I'll talk about these things in separate video. Okay, in details of the type 4 hypersensitivity reactions. Okay, so these were the very basic introductions part about the each individual's the hypersensitivity type reactions. Now I'll focus today about only the type 1 hypersensitivity reactions. Okay, so let's know about type 1 hypersensitivity reactions. Okay, so type 1 hypersensitivity reactions. See, what can you get in terms of the type 1 hypersensitivity reactions? For example, any individual who has gone to the gardens where the there is availability of the pollen grains in the gardens, and this, if we just see that the lungs, the individual just like this. Okay. Now see, within the in the garden he has gone over here, and there is the presence of what, pollen grain. For example, these are the pollen grains. Okay. Now, when there is a chances for the pollen grain to go through the nasal route over here, where within the lungs. Now, just to remove the pollen grains, whatever the mechanisms are present within the lungs, if we see over here, that what, pseudostratified ciliated columnar epithelium, these cells are over here. Now, by the mucociliary activity, okay, by the mucociliary activity, whatever the thing we'll see at first, these cells will try to send back the pollen grains. For example, the pollen grain has come over here. Here the pollen grains. Now these cells will what? These cells will send back the pollen grains from here. So from here the larynx to the pharynx. Then it will come with the esophagus. After that it can go where? Within the stomach. And whenever these pollen grains will be taken by the stomach, we know there is the presence of what? HCl. And this HCl, they will destroy the pollen grains over here. So that's the one thing, okay? The next thing is over here, by chance, the pollen grain has come within the lungs, the inner side of the lungs over here. Okay. Now, whenever the pollen grains will come over here, just try to understand from this thing that these pollen grains. But we know that within the lungs, there is the presence of alveolar macrophage. Alveolar macrophage. Now, this alveolar macrophage, they will just take these all particles within them. Okay, they will take these particles within them. After that, whatever the phenomenon we will see, if I just show you over here these portions, okay, I'm just showing this part only, as you can understand. Okay, let me show only this part. Okay, now whenever the pollen grains will be taken by this alveolar macrophage, this alveolar macrophage, now what they will do, okay, after taking within them, these are the pollen grains. Okay, these are the pollen grains. Now in these cases we will see, after taking within them, they will secrete the lysosomal enzymes. They will secrete the lysosomal enzymes. As a result, we will see there will be complete destructions of the what? There will be destructions of pollen grain. So there will be no symptoms and there will be no chances for this type of the thing. Okay. Pollen grain. There will be no hypersensitivity reactions because the macrophage, okay, this is the macrophage, alveolar macrophage. This macrophage, they will take the pollen grain within them. After that, they will secrete from the lysosomes the different types of the destructive enzymes. As a result, we will see there will be destructions of the what? The pollen grains. There will be no such symptoms in these cases. Now, for examples, one individual is there who is having the bad luck over here. Okay, we are going to talk right now. Now, if we see the lungs of the individual who is having the chances for having this type of the hypersensitivity reactions, now let's talk about them. Okay. It's not anatomically correct, but for understanding purpose, just take it normally, okay? Now see, whenever for example, the pollen grain has come over here, okay, the macrophage, they have stimulated for the productions of the lysosomal enzymes. Now the lysosomal enzymes has degraded the pollen grains partially, not completely. Why? Because whenever the pollen grains will be not will be not destructed completely. As a result, if we see the partial restrictions, we'll see still there will be some left pollen grains over here. Some amount of the pollen grains will be left over here. Because if we see within the macrophage, within the nucleus, within the macrophage, within the nucleus, if we see the structures, in the chromosome number 6, in the chromosome number 6, there will be certain types of the HLA genes. That means HLA genes. HLA, human leukocyte genes over here. These things are like the predominantly genetically predisposition. That means these things are genetically fixed over here, and they will produce these genes. Okay, if we see this type of the genes over here, these genes will produce such types of the MHC2 class molecule, MHC class 2 molecule. And whenever the MHC2 class 2 molecule will be just come over here, as a result, we will see this MHC2 molecule. They will take the pollen grains within them, so there will be no chances for further destructions of the pollen grains. So the MHC2 molecule that will take what? The pollen grains, okay, on the surface of them. Now, if we see the further complications, whatever thing we'll see over here, that the MHC2 molecule, okay, let me show you this one. For example, is the MHC2 class molecule, and they have taken the pollen grains within there on the surface of them. Now they will represent the pollen grains within the surface of the alveolar macrophage. Now whatever the thing we see further, see For these things, let me show a little bit part over here. Let me erase this part. Now see, for example, it's the macrophage over here. Okay. Now in these cases we have seen that the MHC2 class 2 molecules, okay, they have represented the what? They have represented the, okay, let me show you the different color, it will be easier to understand them. It is the class 2 molecule. Okay. Now see, in these cases, if you see that the pollen grains are just presented or represented by these MHC2 class 2 molecule. Okay. Now whenever the MHC2 class MHC class 2 molecule will represent this part over here, and whatever the changes you will see, there will be presence of what? T lymphocyte. There will come out the T lymphocytes. Now we'll see there will be arrival of the T lymphocyte. Now whenever we will see the T lymphocyte will come over here, whenever we will see the T lymphocyte will come over here. Okay. Now within the T lymphocyte we know there is the presence of what? T cell receptors. There is the presence of T cell receptors. Let me show this one only. Now these T cell receptors, they will get connected with the pollen grains, that means within the antigens over here. And within the, we know that within the what? In Th2, that means, for example, in these cases there is a presence of what? Another type of the receptors that are CD4 positive. That means as we know they are CD4 positive, that means there will be presence of what? CD4 receptors. Now CD4 for these things, let me show over here. For example, is the CD4. Now this CD4, they will combine with the what? They will combine with the MHC2 class 2 molecule. That means the MHC2, these things will get combined with the CD4 receptors over here, and the antigens that will combine with what? The antigen that will get combined within the what? T cell receptors over here. Okay. Now after the next things we will see, these things will be sent to the what? Within the lymph nodes. Okay. Now it will be sent to the within the lymph nodes, and after that, whatever the further complications we will see over here. See, is the T cells. Okay. Let me show over here. Okay. From these questions I can show. Now see, whenever these things will get connected, that means the antigens will get connected within the TCR. That means the T cell receptors will get connected with the antigens, and the CD4 receptors over here that will connected with the MHC2 molecules. That means we can write it over here. The T cell receptors that will get connected with the antigens, and the MHC class 2 molecule that will get connected with the CD4. Okay. After that we will see there will be more that will come from here. That will be more co-signals, or you can say co-receptors are here to get the more signals. But on top of the signals we will see under that there is a presence of over here that is known as CD3. Whenever the CD3 will be activated, this domain part will be activated, they will trigger the nucleus. They will trigger the nucleus of the T cells. And whenever the T cells, the nucleus of the T cells will be triggered by these things over here. Okay. After that, whatever the thing we'll see, let me show in this part. So whenever the nucleus that will be triggered by the CD3 over here. Okay. The CD3, for example, one gene will be activated from here. One genes will be activated from these regions, and after the activations they will produce the proteins. And if we know the process, first the messenger RNA, then the protein will come just like this. So one type of the protein they will produce, that is known as interleukin 2. We'll get what? Interleukin. Okay. And whenever the interleukin 2 will be produced, after that we'll see there will be activation of another type of the gene over here, and this gene that will produce another type of the protein that is known as interleukin 2 receptors. This is the interleukin 2 receptors. Now the interleukin 2 will come over here. If you see the interleukin 2, if we see over here, the interleukin 2 that will come over here, and whenever the interleukin 2 will get connected with the interleukin 2 receptors, we'll see there will be activation of the another type of the, because the signal will go within the nucleus, and we'll see activation of another type of the gene. And whenever this type of the gene will be activated, this will produce what? This will produce interleukin 3. Sorry, interleukin 4 over here. Yes. So interleukin 4. And whenever the interleukin 4 will be activated, this will lead to the clonal formations of the T cells. We will see over in these cases there will be clonal formations of the T cells. That means we will see what? There will be too much amount of the T cells, just like this. So we can say right over here the clonal expansion. It's better word we can use over here, clonal expansion. So we will get over here the clonal expansion. Now when you get clonal expansions, now I'll just tell you another thing over here. Whenever we are getting the pollen glands, whenever it is going to the lymph nodes, there is the presence of the B lymphocyte as well. Now for example, is the B lymphocyte over here. Okay. Now within the B lymphocyte there are some receptors responsible for taking the pollen grains within them. Okay. See, so is the pollen grain, and whenever the pollen grain that will be connected with the B cell receptor, so what is the BCR, that means B cell receptor, and they will send the signals within the nucleus of them. Whenever the nucleus that will be activated, the signal, they will see the nucleus activating the genes. From here we will get another type of the gene activation. So whenever the gene that will be activated, that will produce certain types of the proteins. And for example, they are producing the protein that is, let me show in this way, that is interleukin 4 receptors. What? Interleukin 4 receptor. So whenever the interleukin 4 receptors will be here, because I have already said there is the presence of interleukin 4. Now within the T cells there is already presence of what? Interleukin 4. So interleukin 4. Interleukin 4 over here. Now this interleukin 4, they will go where? They will go within the B cells and they will just do the activations of the B cells. And whenever the B cells that will be activated over here by the stimulations of the interleukin 4, we will see that there will be also the clonal expansions of the B cells. We will see the clonal expansion of the B cell. We will get the clonal expansion of the B cell. And in these cases, whenever we are getting the clonal expansion of the B cell, just try to understand from this thing that there is already presence of what? Interleukin 4 receptors. Okay. Then after the activations of the interleukin 4 receptors, we will get the clonal expansion of the B cells. After that, we will get over here another receptors, that is for interleukin 5. That is for interleukin 5. Now see, one thing over here, whenever I have said over here interleukin 4, after that another type of the gene that will be activated within the T lymphocyte, that is known as what? Interleukin 5. Interleukin 5 over here. Now for example, there is what? Presence of interleukin 5. Now there is the receptors for what? Within the B cells for interleukin 5. Interleukin 5. Now interleukin 5 is over here in all of the cells. Now this interleukin 5 that will go where? That will go in these regions, because the receptor for the interleukin 5. Now whenever the interleukin 5 will come over here, and that will convert the B cells into plasma cells. That will convert the B cells into the plasma cells. And whenever we will get the plasma cells over here, in this cases we will get the cartwheel shape of the nucleus just like this. Along with that we will get Golgi apparatus, condensed Golgi apparatus. After that we will see these plasma cells that will get converted into what? That will produce the antibodies. And what type of the antibodies they will produce over here? IgE. And another thing that whenever we are getting the interleukin 4, try to understand, whenever we are getting the interleukin 4, this interleukin 4 can stimulate also the productions of the IgE. This is responsible also for producing more amount of the IgE antibody. Okay. So in this portions we got what? In this portions we got there is a productions of the interleukin what? IgE antibody. Okay. Whenever the IgE antibody will be produced, now this IgE antibody, where they will go? They will go in the mast cell, within the tissue, and on the basically within the circulations. Now for example, if I want to show you that after productions of the IgE antibody is the mast cell over here, and within the mast cell we will see there is the receptor for the IgE antibody. Now we will get over here the IgE antibody. They will come over here. We'll see what IgE antibody in these cases. Now in the further cases we will see whenever the individual will get affected again the pollen grain in these cases, then these pollen grains will come over here and they will cause the further damage the tissues. So this type of the thing we will just talk about in the next part of the video. So I hope you understood how the Ig antibody is coming. Okay, whenever the pollen grains are coming within the body and how they are activating the different types of the immune cells for productions of the Ig antibodies. Okay, so see you in the next video. About the further complications from the mast cell, we will talk about what type of the different types of the restrictive enzymes are secreted and how they are affecting the different tissues and the different organs. Okay, so see you in the next video. Thanks for watching.